Hi student, this is Professor Harmony Kim, and this video is lecture for tissue. What is the definition of tissue? Tissue is a group of cells of a similar structure that perform a common function. So tissue is mass of like cells. The cells in a tissue share morphological features. Therefore, disruption of the structure is a sign of injury or disease. Such changes can be detected through histology. Histology is an academic field to study microscopic study of tissue appearance, organization, and function. There are 4 basic types of tissue: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. What is the criterion to determine some group of cells belong to each type of tissue? It is based on embryonic origin. The cells within each type of tissue share a common embryonic origin. If we look at this right panel, here's egg and here's sperm, and they fuse, fertilize to form zygote. Zygote is a single cell. Then these zygotes undergo mitotic division actively and differentiation and develop into a new organism. As meiosis goes on, the lineage of embryonic cells form the distinct germ layers, and from each germ layer, all the tissues formed. If each germ layer is located in outer position, it is called ectoderm. If each germ layer is located in middle portion, it's like a pink color, it is called mesoderm. And if each germ layer is located in inside inner portion, then it is called endoderm. And this figure shows the type of tissues associated with each of the 3 germ layers. So in case of ectoderm, the nervous tissue derives primarily from the ectoderm, and muscle tissue derives from mesoderm, and epithelial tissue originates in some of all 3 layers. Some of endoderm, some of mesoderm, and some of ectoderm become epithelial tissue. This left panel shows that the blue color, the outer layer, indicate ectoderm, and in the middle, red color indicate mesoderm, and epithelial tissue can be formed, some of endoderm, some of mesoderm, and some of ectoderm. They all together participate to form epithelial tissue. What are the functions of epithelial tissue? Epithelial tissue covers exterior surface of the body, and epithelial tissue line internal cavities and passageways and form certain glands. So, one of the functions of epithelial tissue is protection, because it covers body and lines all body cavities. Therefore, epithelial tissue provides a protective shield for underlying tissues. For example, your skin. Epithelial tissues provide the body's first line of protection for physical, chemical, and biological wear and tear. Do you think there are many bacteria in the air? why those bacteria cannot enter your body. Is it easy for bacteria to enter body? Because of skin, they have a hard time to enter. Do you know why burning is dangerous? Once your skin is burned and then your epidermis peeled off, then bacteria in the air easily sitting on that damaged burning skin, and then they can enter your body easily. In other words, epithelial tissue provides the body's first line of protection. The cell of an epithelium acts as gatekeeper of the body, controlling permeability and also allowing selective transfer of materials across a physical barrier. All substances that enter the body must cross an epithelium. Even when you eat, when you breathe in, all substances need to enter the body through epithelium. Second function is absorption. Epithelial tissue absorbs nutrients in some part of the body. For example, digestive tract, in intestine, then there's epithelial cell, and then epithelial cell absorb nutrient. Third function is secretion. Epithelial cell is specialized to secrete certain product in some part of the body. For example, cells lining the respiratory tract secrete mucus, and this mucus traps incoming microorganisms and dust particles. Some epithelium of small intestine release digestive enzyme. So epithelial tissue has capacity to secrete. Another function is filtration or excretion. Epithelium in the kidney cell get rid of the body of material such as waste. Therefore, epithelium role as a passage of a small molecule through a sieve-like membrane. Sieve-like, S-I-E-V-E, sieve-like membrane, kinds of filtration. So in kidney, water can be absorbed and waste substance can be removed. Another function of epithelial tissue is sensory reception. For example, the taste bud in the mouth allow you to have a sensory function. There are several types of epithelial tissue. We will study a little bit later, but they all share unique characteristics of epithelial tissue which are common to most types. First is cellularity. Epithelium consists almost entirely of cells. Epithelium has very little extracellular matrix between them. So if you look at this epithelial cell here, between cell, there's a very little extracellular space. Second characteristic is specialized contact. Adjacent epithelial cells are directly held together by cell junction. For example, tight junction, adhesive belt, desmosome, gap junction. These kind of cell junction make epithelial cell held together very closely. Polarity means most epithelial tissue have one free apical surface not attached to other cells. So this part is apical surface and the other side is basal surface. This apical side, the free surface, open lines lumen of duct vessels or lumen of a cavity. The basal surface of most epithelial tissue is attached to a basement membrane. Basement membrane is meshwork of protein and fiber. The basement membrane plays an important role in supporting and guiding cell migration during tissue repair. Epithelial tissue does not contain blood vessel. Look at this is epithelium and bottom part is connective tissue. Connective tissue has supply of blood vessel, but this blood vessel cannot enter the epithelial tissue. So epithelial tissue does not contain blood vessel. Lack of blood vessel, we call that avascular. A means no. So capillaries in connective tissue do not penetrate to the basement membrane. The last characteristic of epithelia is regeneration. Epithelium rapidly replaces lost cells or damaged cells by mitosis. Epithelia are classified according to two criteria. First, number of layers. In other words, the relative number of layers. Some epithelial tissue has only single layer. And second criteria is shape of the cell. So these two criteria generate the name of types of epithelial tissue. But first you have to bring the number of layers. If epithelial tissue is composed of a single layer of cells, then you will just say simple. If epithelial tissue has more than one layer, then you will put stratified epithelia. Stratified epithelium consists of more than one layer of epithelial cell. In other words, in stratified epithelial tissue, some cells sitting on the top of other cells. Now, first you have to bring the number of layer of cell, and that word followed by cell shape. So cell shape is the other criteria to classify epithelial tissue, and this criteria is based on cell shape. First, squamous means flat and thin. Look at here, very thin. It's like disc-shaped nuclei. And another cell shape of epithelial tissue is cuboidal. Cuboidal, look like a cube shaped like a disc. In other words, height and width almost the same. And nucleus located in a center. Columnar is height is more than width. Look like a column. And the nucleus is located not in the center, but close to basal surface. When epithelium has several layers of epithelial cell, like this, it could be mixed with the top portion is a squamous epithelial cell, and the lower portion is cuboidal epithelial cell. Then how you can name this tissue? Then you have to choose one kind of cell shape for the naming. That is, you have to look at the top portion. So the stratified epithelia are named according to the shape of the cell in the apical layer. Even though the basal portion of this epithelium consists of cuboidal epithelium, still we can put these epithelial tissue as stratified squamous. So stratified means it is composed of several cell layers. And several cell layers can be constituted with different kinds of cell shape. But you have to choose only top portion. So stratified and followed by the shape of cell of apical region. Therefore, squamous. So far, we look at squamous, cuboidal, and columnar epithelial cell, but there's some two other types of classification of epithelia. One is pseudostratified columnar epithelium. Pseudo means fake, not true. In other words, looks like several layers, looks like more than one layers, but actually that is not true. That means single layer. And this pseudostratified epithelial cell occur columnar epithelium. So single layer of cell, and some cells reach the free surface, this apical side. Some other cells do not reach the apical side. This gives false impression due to different nuclei position. Look at here. Look like this is one layer of nuclei, and look like this is another layer of nuclei. So you think that, okay, this is one layer of epithelial cell. This is another layer of epithelial cell. So it looks like stratified epithelium. But that's not true. If we look at this cell, the top portion cannot reach the apical side. Also look at this one. Also maybe this one. This one you can see the basal part is attached to basement membrane, but the other side of this cell cannot reach the apical region. Therefore, if we look at closely, it is not more than one layer of epithelial cell. It is just one layer. So we put that look like stratified, but actually not. Therefore, we call this pseudostratified columnar epithelium. And this pseudostratified columnar epithelium is usually ciliated and secrete mucus. So on the top of this apical region, they have whip-like or short hair-like cilia. Cilia is attached. Another type of classification of epithelia is transitional epithelium. Transitional means, especially this apical region, epithelial cell change the shape of a cell. Right now, you look like cuboidal epithelial cell. But when the organ is stretched, then this cuboidal epithelial cell change shape to squamous epithelial cell. Now, this organ is not stretched, then squamous epithelial cell go back to original shape, such as cuboidal epithelial cell. This transitional epithelium undergo transition in shape. Therefore, we call this kind of epithelium transitional epithelium. Urinary bladder has transitional epithelium so that urinary bladder can accommodate fluctuation in the volume of urine. So far we study simple epithelia or stratified epithelia. And in case of a simple epithelia, there's a simple squamous, simple cuboidal, simple columnar epithelia, and also pseudostratified columnar epithelia belong to simple epithelial category. Stratified squamous can be further subcategorized: stratified columnar, stratified cuboidal, stratified squamous. Transitional epithelia, of course, it has several layers, but we do not put stratified transitional epithelium. We just call transitional epithelium. So we will study one by one in detail. And if you look at this diagram, you can feel that the structure of epithelium reflects its function. Simple squamous epithelium function for diffusion and filtration, and stratified squamous epithelium is for protection. So sometimes maybe you can see your skin can be peeling up, but you do not breathing. Why you do not breathe? Because there is no blood vessel run through epithelial tissue. So stratified squamous epithelium is for protection. Simple cuboidal and simple columnar epithelium function for secretion and absorption. And stratified cuboidal and stratified columnar epithelium function for protection. So we will study each epithelia in detail one by one. First, simple squamous epithelia. Single layer of flat and thin epithelial cell. The nuclei appear as a bump. Can you see like this? This is cross section. Nuclei appear as a bump when viewed as a cross section because the cells are so flat. In surface view, you can see the free surface and nucleus. Here's nucleus. And when you look at the between cell, you can see very little extracellular materials between the cell. So cell to cell is almost closely held together. Because of the thinness of cell, simple squamous epithelia is present where rapid passage of chemical compounds take place by way of diffusion and filtration. Some simple squamous epithelia make a very smooth surface by secreting lubricating substances. We study serosa membrane. Serous membrane secrete lubricating substance so that they wanna reduce friction or abrasion. That kind of secretion can occur by simple squamous epithelia. Simple squamous epithelia can be found in alveoli of the lung where gas exchange occurs. So gas can easily diffuse in and out. Therefore, they need very thin cell. So simple squamous epithelia can be found in alveoli of the lung. Alveoli is the terminal region of lung, and capillary blood vessels surrounding these alveoli, and then gas diffusion can easily take place. Simple squamous epithelia also can be observed in lining of capillary blood vessel, also lining of heart and lining of lymphatic vessels, which is called endothelium. Endothelium, endo means internal covering. Also, simple squamous epithelia can be found lining of serous membrane of peritoneal, pleural, and pericardial cavities, which is referred to as mesothelium. Meso means middle covering. The mesothelium is a simple squamous epithelium that forms the surface layer of serous membrane that lines body cavity and internal organs. So, where you can easily observe simple squamous epithelia in human body, maybe some portion of kidney tubule and maybe inner surface of eardrum. And also we can observe simple squamous epithelia in alveoli of the lung or lining of blood vessel, lining of serous membrane. Another type of epithelial tissue is simple cuboidal epithelia. Cuboidal epithelia. So cell has shape, cube-like shape, and simple, therefore single layer of cube-shaped cells. And the size of a nucleus is large and located at the center of the cell. Simple cuboidal epithelia are active in the secretion and absorption of molecules. These epithelia are observed in the lining of kidney tubule and kidney gland because wastes are secreted and waters are observed in the kidney. Therefore, simple cuboidal epithelia are found in the kidney. So here's microscopic view, cube-like, and then you can see large size of nucleus. And here's basement membrane. We will study that a little bit later, but epithelial cell will receive support from connective tissue. So there is some between epithelial tissue and connective tissue, there is basement membrane. So simple cuboidal epithelia, how it look like, and then based on that structure, what are the function, secretion and absorption, and then where you can find simple cuboidal epithelia in human body, mainly kidney region. Because in kidney, many secretion and absorption take place. I think this figure is from the text, so you can review this. The more you see the figure, the more you get familiar with each type of epithelial tissue. So here's a kidney. So kidney tubule, you can see, look at here, each cuboidal shape and then single layer. Therefore, we call this simple cuboidal epithelia. So in kidney region, we can observe simple cuboidal epithelia. Kidney is composed of a convoluted tubule, collecting duct, ascending loop of Henle, surface of ovary, or thyroid gland. Those are the places we can observe simple cuboidal epithelia. Another type is simple columnar epithelia. Still single layer of tall, narrow cell. So tall, narrow cell look like a column. And if we look at the position of a nucleus is elongated and clustered at the basal end. In other words, the cuboidal, look at here, is round, big, and located at the center, but columnar epithelia, the shape of a nuclei is elongated and also their position is more clustered at the base end. Some cells have cilia, or some columnar epithelia has microvilli. Cilia and microvilli, they are different. Simple columnar epithelia has goblet cell. Goblet cells secrete mucus. If you look at this part here, goblet cell look like champagne glass. So I will show that a little bit later, but these simple columnar epithelia or pseudostratified columnar epithelia, they have goblet cell. This goblet cell is kind of integrated throughout simple columnar epithelia. And this goblet cell secrete mucus. And once mucus produced, then surface transport with cilia. Some columnar epithelia has cilia. Therefore, these cilia move in one direction and propel mucus. Sometimes some simple columnar epithelia with cilia can be observed in uterine tube, and that cilia move oocyte through uterine tube. Some simple columnar epithelia has microvilli instead of cilia, and that kind of columnar epithelia have microvilli can be observed in intestine. Then that intestine simple columnar epithelia secrete some digestive enzyme, or also then microvilli role as absorb some nutrient. So let's look at microscopic view. Again, single layer of tall, narrowed cells. Some cells have cilia, some cells have microvilli. In this microscopic view shows microvilli look like brush border, look like microvilli. Therefore, simple columnar epithelia have microvilli can be observed in intestine. Okay? And what do? Is they secrete some enzyme or they absorb nutrient. In this slide, there is no example of simple columnar epithelia with cilia, but you can imagine that the cilia is a little bit longer than microvilli. Then simple columnar epithelia with cilia can be found in the lung and uterine tube or uterus and auditory tube. Simple columnar epithelia with cilia, those cells participate in movement of a particle out of the bronchial of the lung by ciliated cell. Also, simple columnar epithelia partially responsible for the movement of oocyte through the uterine tube by ciliated cell. Okay? So, this is the region where we can observe simple columnar epithelia with cilia. Also, ventricle of brain is another location we can observe simple columnar epithelia with cilia. Ventricle is the space in the brain, and some fluid run through this ventricle, and that fluid is cerebrospinal fluid, and therefore this is kinds of a conducting space, and that space is constituted with simple columnar epithelia with cilia. Columnar epithelia, but this time is pseudostratified columnar epithelia. Still single layer. I said this pseudo means look like 2 layer or 3 layer, but actually single layer of cells, not stratified. And position of nuclei is varies. As I said, some cells are tall and thin and reach the free surface, and some other cells do not reach the surface. Single layer of irregularly shaped cells and differently sized columnar cell. This one and then this one, this cell is larger than this cell. So the size is different, and also the columnar distribution is not regular shaped cell. These columnar epithelia are not shaped regularly. The nuclei of these cells are at different level and appear stratified, but actually single layer of cell because all the cells are in contact with basal lamina, although some do not reach the apical surface. So when you look at bottom, each cell touch lamina or basement membrane. But even though they touch basement membrane, some cells cannot reach the top apical region. Pseudostratified columnar epithelia almost always ciliated, here cilia, and associated with goblet cell. Here's you can see blue color. These are the goblet cells. These goblet cells secrete mucus. So, pseudostratified columnar epithelia and simple columnar epithelia both have goblet cell. Therefore, these two types of epithelia participate in secrete mucus. And simple columnar epithelia usually found in bronchial of lung and uterine tube and intestine, but pseudostratified columnar epithelia mainly can be found in this trachea region. So, lining of nasal cavity, nasal sinus, auditory tube, pharynx, trachea. This is the main region we can observe pseudostratified columnar epithelia. Since it's almost always ciliated, look at here, this ciliated, therefore this ciliated form lines trachea and most of upper respiratory tract. So here, cilia is longer than microvilli. Now let's move to stratified squamous epithelia. Several layers of squamous epithelia at the top surface. If we look at this stratified epithelia, multiple layers of cells that are cuboidal epithelia in the basal layer and progressively flattened toward the surface. So, two types of epithelia mixed to form this tissue. Then how you can call this tissue? First, stratified. Then you will look at the apical region is composed of squamous epithelia. Therefore, the name is stratified squamous epithelia. This epithelium can be non-keratinized or keratinized. It could be keratinized or it could not be keratinized. In case of non-keratinized stratified squamous epithelium, the surface cells retain a nucleus and cytoplasm, so there's some kinds of metabolism continue to take place. But some stratified squamous epithelia is keratinized. Then the cytoplasm of cells at the surface, especially apical region, is replaced by a protein called keratin, and those cells are already dead cells. What is the function of stratified squamous epithelia? Protection against abrasion, a barrier against infection, reduction of water loss. Where you can find stratified squamous epithelia which carries this function. Good example is mammalian skin. Mammalian skin is an example of dry, keratinized, stratified squamous epithelium. So, stratified squamous epithelium can be observed in skin and scalp and sore. I said there's some non-keratinized stratified squamous epithelia. Those tissues can be observed in mouth, throat, esophagus, anus, vagina, and cornea. So inside the mouth, inside the cheek, you have stratified squamous epithelia, but they are not keratinized. So the lining of a mouth cavity is an example of unkeratinized stratified squamous epithelia. You can see that here several layers of cell, and each cell shows flat, thin shape. So these are squamous epithelia, and there are several layers, therefore stratified squamous epithelium. The nucleus is flat, okay, and here is connective tissue and epithelial tissue. On the border, there is a basement membrane. Let's move to stratified cuboidal epithelia. Multiple layers of cube-shaped cells. What are the main functions? Secretion and absorption. Also protect against infection. So if you look at this microscopic view, you can see this cuboidal one layer and another layer. And here's a free surface, and this indicates nucleus, and then this, this indicates nucleus, and this indicates basement membrane. Here, this line indicates basement membrane. Where stratified cuboidal epithelia are observed, the location is sweat gland duct, mammary glands, and salivary gland duct. So inside the mouth, there's 3 places when saliva secreted. One is parotid gland, one is sublingual gland, another one is submandibular gland. These glands are composed of stratified cuboidal epithelia. So 2 layers of cuboidal epithelia, and microscopic view shows 1 layer, 2 layer. So we call this stratified cuboidal epithelia, where you can observe stratified cuboidal epithelia, sweat gland duct, mammary glands, salivary gland duct, because its function is secretion and absorption. Stratified columnar epithelia composed of a multiple layer of a cell with tall, thin cells resting on layers of more cuboidal cells. So if we look at here, this is this layer close to free surface, that is columnar epithelia. But the basal portion is more like cuboidal epithelia. So cuboidal cells at basal layer and tall and thin layer on the top surface. What is the function? Protection and secretion. And where you can find stratified columnar epithelia, a portion of male urethra. So stratified columnar epithelia is not widely distributed in human body. It's very rarely distributed. So you can see a portion of a male urethra, we can observe stratified columnar epithelia. Again, here's epithelia, and here's connective tissue. The borderline, we call that basement membrane. Transitional epithelia. Of course, it belongs to stratified epithelial category, but we do not put simple or stratified in front of transitional epithelia. We just more focus on changing the shape and transition of shape. Therefore, we just call transitional epithelia. And it is composed of several layers of epithelial cell. When organs is not stretched, cells appear as cuboidal epithelia. When the organ is stretched, cells appear squamous epithelia. Here, the organs is not stretched, therefore you can see cuboidal epithelia. Once this organ stretch, then this cube shape changes squamous epithelial cell. Where you can find the transitional epithelia? Lining of a urinary bladder. This is a urinary bladder. And ureter is this one. And superior urethra is this part. The location related to urinary bladder, they have transitional epithelia. What is the function of transitional epithelia? It permits distension of a urinary organ by contained urine. So once again, this microscopic view shows you can see dome-like cuboidal shape to squamous epithelium when the bladder is empty. Then these apical cells undergo transition from cuboidal to squamous as bladder fills with urine. Therefore, transitional epithelia is found only in the urinary system. Simple columnar epithelia and pseudostratified columnar epithelia, they have goblet cells. Goblet cell is unicellular. Look at here, just a single cell. And observe in the epithelial lining of intestine and respiratory tract. Goblet cell produce mucin. Mucin is glycoprotein. When this glycoprotein mucin dissolves in water, it becomes slimy mucus. Goblet cell contain abundant organelle such as ribosome, Golgi apparatus, endoplasmic reticulum. Those are the responsible for the synthesis and secretion of mucus. And you can see many secretory vesicles filled with mucin. Mucus covers, protects, and lubricates the internal body surface For example, a large amount of mucus protects the stomach lining against hydrochloric acid produced in the stomach. Therefore, goblet cell can be observed in respiratory tract or intestine. And goblet cell can be present amid of simple columnar epithelia or pseudostratified columnar epithelia. It looks like champagne glass, that's why this cell has name goblet cell. We are studying epithelial tissue. A group of epithelial cells is a little modified. So, granular epithelial is called gland. Most glands consist of a group of epithelial cells. Gland is a secretory structure made up of one or more cells modified so they can synthesize and secrete chemical substances. Gland cells obtain substance from the blood. Can you see this blood in capillary? And gland cells convert the substance received from the blood into a product and secrete aqueous fluid that usually contain protein. And they secrete to blood. So glands are classified by the site of release, in other words, where the gland released the product. Based on that criteria, glands can be classified endocrine gland or exocrine gland. This left panel shows endocrine gland, right panel shows exocrine gland. Endocrine gland is a ductless gland. This part we call a duct, D-U-C-T, and then there is no duct. So we call this a ductless gland. And this ductless gland releases secretions directly into surrounding tissue or fluid. So in this figure, the substance released to the blood. Exocrine gland secrete the product through the duct. Through the duct, there is opening. Therefore, the product can be released to the external environment. Endocrine glands is called ductless gland. The product secreted by endocrine gland are called hormones. These hormones are released into extracellular space, just the surrounding tissue, and then enter the blood vessel and travel through the bloodstream to deliver chemical message to specific target organ far from the site of release. The example of endocrine glands include anterior pituitary gland, thymus, adrenal gland, and endocrine glands in the intestine. For example, endocrine glands in the intestine secrete a hormone that signals the pancreas to release the enzyme that help digest a meal. Most glands are multicellular, consisting of epithelium and the supporting network of connective tissue. Exocrine glands have a duct and secrete the product into a body surface or body cavity through a duct. What exocrine glands secrete are mucus, sweat oil, or saliva, or breast milk. Exocrine glands are classified by the number of cells forming the gland. So exocrine gland can further subcategorize based on the number of cells forming the exocrine gland. One is unicellular exocrine gland, The other category is multicellular exocrine gland. The example of unicellular exocrine gland is Goblet Cell. Do you remember? It looked like a champagne glass and it has opening to exterior environment. So, the example of a goblet cell which is scatters in the epithelial lining of intestine and respiratory tract between simple columnar epithelial cell, that is the example of a unicellular exocrine gland. Multicellular glands have two basic parts. One is epithelium walled duct and the other part is a secretory unit. Where is this part? Before we look at the diagram, so multicellular exocrine glands has two parts, epithelium walled duct and secretory unit, and also the structure of a duct further classified the gland into simple duct, compound duct. In case of a simple duct, it does not have a branched duct, but compound duct has a branched duct. And another classification based on secretory unit, if secretory cells form tube, then we call that tubule. If a secretory unit form like a pocket, then we call that alveoli. And tubular alveoli means contain both kinds of secretory unit. Okay, we will see this diagram. So here's simple duct and compound duct. And if we look at the color, can you see dense yellow color? Is yellow color this area? We call this duct. Here's duct. Here's duct, duct, duct. And if we look at this red color portion is secretory epithelium. In this compound duct has several secretory epithelium. Simple duct has single secretory epithelium. So each multicellular exocrine gland has two basic parts. One is epithelium walled duct, look like yellow color, and secretory unit consisting of secretory epithelium. Here, this is epithelium, but they secreting substance. Every duct has that two portions. If we look at further, the structure of a duct further classified the gland into simple duct and compound duct. Simple duct means the duct, this yellow portion, is not branched. So duct is single. If we look at these three secretory epithelium, you might think that, oh, the duct might branch it. No, look at all these yellow portion, still one. Here, like a tube shape of secretory epithelium. But when you look at duct, it's still one. We call this simple duct. What about compound duct? Just on the top of secretory epithelium, this yellow portion, this is a duct. This is duct. This is duct. Maybe originally it was single duct, but this duct branched. Here, here, here. We call this compound duct. Okay, there's further classification can be done based on a secretory unit. Look at only red portion. What kind of shape? Like a tube shape. We call that tubular. Also tubular, but branched. Therefore, we call this simple branched tubular exocrine gland. Some secretory epithelium has round pocket. It's not tuber. Therefore, we call this simple alveolar exocrine gland. Simple branched alveolar, exocrine gland. What about here? It's a compound. When you look at these, look like tube shape. Therefore, we can call compound tubular exocrine gland. But if we look at here, this is compound alveolar exocrine gland. And in this exocrine gland, kinds of a mixture with alveolar and like this tubular. So compound tubular alveolar exocrine gland. So salivary gland has this exocrine gland, and mammary gland, secrete milk, has this kind of structure. Okay, so there's some example how each gland has a different structure. You can just look at that. I will not ask about what kind of gland you can find in a stomach or maybe in mammary gland. I will not ask about that question on the exam, but I might ask about how can you call this? Then you can think about, okay, just single unbranched duct, therefore simple. And then look at this secretory epithelium. It has a tube structure, so you can say simple tubular exocrine gland. Okay. Same thing, but when you look at this duct area, it's branched, therefore compound. And what kind of secretory epithelium shape? Is still tubular. So compound tubular exocrine gland. So I just want to know whether students understand how to put the naming. Let's look at epithelial cell. We said this is apical region, this is basal region, and then let's look at the side, lateral surface. So lateral surface feature of epithelial tissue. Cells of epithelia are closely connected and they are not separated. Epithelial tissue is not really tough and strong, but how they can hold together each other. Several types of connection allow some degree of interaction between the cells. Epithelial cells are connected to one another in three ways. One way is adhesion protein in the plasma membrane of the adjacent cells linked together in the extracellular matrix. So what will be adhesion plant? We will study that a little bit later. Or the wavy contour of the membrane of adjacent cells join in tongue and groove fashion. So they can just fit each other very well by way of some membrane protein. Or there's special cell junctions. All these factors make epithelial cell put together. So we will study special cell junction more detail. One of cell junction called tight junction, jonula occludens. Some proteins in plasma membrane of adjacent cells fuse together so tight, so there is no any small extracellular space between two cells. Tight junction close off extracellular space. Therefore, tight junction prevent any molecule from passing between the cells of epithelial tissue. So when two adjacent epithelial cells form a tight junction, there is no extracellular space between them, and the movement of substance through the extracellular space between the cells is blocked. This enables the epithelia to act as a selective barrier. Tight junction can regulate what material cross the epithelial layer. Tight junctions can be found in the lining of intestine. Also, in the brain, there's blood-brain barrier. Blood-brain barrier has a tight junction so that the solute from blood cannot cross into the extracellular fluid of a central nervous system where neurons reside. So, inside the brain, the cortex must be very clean. Another type of cell junction is adherence junction. We call that jonular adherence, a type of anchoring junction. Transmembrane protein attached to actin microfilament in the cytoplasmic portion and then bind adjacent cell. In other words, adjacent cell also has the same situation. Then here's adherence. This transmembrane protein bind to adjacent cell from one side of transmembrane protein. So in other words, one side of transmembrane protein bind to adjacent cell through membrane, and the other side of transmembrane protein attached to actin microfilament in the cytoplasmic portion. And the other cell also has the same situation. So the relay of actin microfilament throughout cytoplasm between cells form kinds of this adhesive belt. Can you see? Here's adhesive belt. This is the example of adherence junction. Together with the tight junction, these form the tight junctional complex around the apical lateral borders of epithelial tissue. So the close to apical region, this is a tight junction. Below to tight junction, it shows adherence junction to form adhesive belt. And how this adhesive belt can be formed? Transmembrane linker protein bind to next cell throughout membrane, but the other side of this transmembrane protein attached to actin microfilament in the cytoplasm, and this actin microfilament relay. Here's a relay. Therefore, it forms like convening belt. We call that adherence junction. Another type of cell junction is a desmosome. Desmosome, this is the main junction for binding cells together. Of course, tight junction and adherence junction, they also contribute to cell junction, but desmosomes is main junction for binding epithelial cells together. If we look at this desmosome complex, you can see this plaque. This plaque, circular plaque on the cytoplasmic face of each plasma membrane. This plaque, face of each plasma membrane. This is plasma membrane. Here's one cell. Here's another cell. And this plaque joined by linker protein. This blue color is called linker protein, named cadherin. This cadherin interdigitate like teeth of a zipper in the extracellular matrix. So one cell, one epithelial cell has plaque, and this plaque attached to cadherin through plasma membrane. And the other cell also same situation. The other cell also has a plaque. This plaque also has linker protein named cadherin. And this cadherin is going to interdigitate. So cadherin is embedded in this patch plaque and project through the cell membrane to link with cadherin, molecule of adjacent cells. In other words, cadherin interdigitate like the teeth of a zipper in the extracellular matrix. This really provides strong strength to bind each other. And this is one side of plaque. The other side of plaque are bind to, this time, intermediate filament. Intermediate filament is one type of cytoskeleton. Okay? Therefore, intermediate filament also support this plaque. So this kind of configuration influence the shape and folding of the epithelial tissue. One more type of cell junction named gap junction. There is a gap. Okay? The cell to cell, they closely bind together, but there is some kinds of a channel is formed, and therefore, even though the cells stay side by side, there are some substances can exchangeable. So, gap junction forms an intercellular passageway between the membrane of adjacent cells. This facilitate, this make easy the movement of a small molecule and ions between the cytoplasm of adjacent cells. So all these kinds of junctions allow epithelial cells can hold together each other. I mentioned several times about basement membrane, and here's epithelial tissue. And then epithelial tissue receives support from connective tissue. And then there is a border between epithelial tissue and connective tissue. We call this basement membrane. The border between epithelium and connective tissue. And supporting sheet, thin non-cellular sheet, like this. This basement membrane is like a sheet. But these are not the cell. These are composed of protein. Those proteins secreted by epithelial cell and some kinds of fiber, reticular fiber, secreted by connective tissue. Those molecules get together to form non-cellular sheet we call basement membrane. Basement membrane acts as a selective filter. It regulates which molecule from capillaries in connective tissue enter the epithelium. Capillary blood vessel cannot penetrate into epithelial cell, but some molecules in capillary can provide those molecules into epithelium. At the time, they need to pass through basement membrane, and sometimes some molecules cannot even enter epithelial cell. So this basement membrane role as a selective filter. And basement membrane is formed basal lamina and reticular fiber. Basal lamina secreted from epithelial cell, and this reticular fiber were produced by connective tissue. Therefore, we put them together called basement membrane. So, basal lamina is portion of a molecule secreted from epithelial cell. Let's look at apical surface feature of epithelial tissue. I mentioned that here's cilia, and then here's microvilli. They are not same. How they are different? Microvilli is a little shaggy hair, finger-like projection. The membrane protrude. so that they can increase the surface area across which small molecules can enter or leave cells. Or also it's very advantageous to absorb nutrients because surface area is increased. So this kind of microvilli can be found in the epithelia of small intestine and also kidney. When you look at cilia, look at the shape. It's longer than microvilli, but the thickness, microvilli is thicker than cilia. In other words, the cilia is longer and thinner. And then what kind of molecule compose cilia? Do you remember? One of cytoskeleton. What is that? Microtubule makeup of cilia. What about microvilli? It's just membrane protrusion. There's no cytoskeleton to form microvilli. Also, cilia is motor. It can move over. Therefore, cilia can propel mucus in one direction. Microvilli is not motor. It's not moving. Just stay there and then just absorb or secrete. And cilia cannot absorb any substance. It just propel. It just move substance in one direction. But microvilli can absorb nutrient. Where can you find cilia? Lining of trachea or bronchi. They produce mucus. Therefore, cilia sweep the mucus. Where microvilli often found? This found in intestine. Therefore, microvilli absorb nutrients. So there's a difference between cilia and microvilli. Please do not confuse. Here's a cilia. Okay, cilia, whip-like, highly motor extension of apical surface membrane. And cilia beat in unison. It's like an oar or canoe paddle. And then they beat only one direction. So the cilia move in coordinated waves, so the wave can push in mucus over the epithelial surface. If you look at the structure inside the cilia, it is composed of microtubule. At the center, you can see two microtubules, and two microtubules surrounded each pair of microtubule. Outside, about 9 pairs of microtubule surround one middle pair. Cilia originate as their microtubule assembled around centriole. Do you remember centriole? 2 centrioles make up of centrosome, and centriole and centrosome actually involved in cell division. But centriole is the place microtubule manufactures. Therefore, cilia originate as their microtubules assemble around the centrioles, then migrate from centrosome to the apical plasma membrane, and then cilia is going to stay on the apical side of epithelial cell. Another extremely long cilium is called flagellum. And flagellum has a little bit different movement compared to cilia. It like a propeller. It rotates in corkscrew motion. And flagella can be found only in sperm cell. So far, we study epithelial tissue. You need to know about how you can naming based on the structure and then what kind of a function each type of epithelial tissue have, and then where you can find each type of epithelial tissue in human body. Okay. Now, I'm going to switch gear to connective tissue. Connective tissue is quite different story from epithelial tissue. So why don't you take a short break, and then I'm going to make connective tissue in another video. So you can watch separate video for connective tissue. So this is the end of epithelial tissue recording.